Thank you very much, Steve. It's a pleasure to be able to do this event with you and Ned and our colleagues from CINJ and George Mason. So it's my pleasure now to introduce Emanuel Petricoin, known as Chip, and Lance Liotta, who are co-directors of the Center for Applied Proteomics and Molecular Medicine at George Mason University. Prior to George Mason, they were at the National Cancer Institute for many years, where they were colleagues of Dr. Labudis. And since arriving at George Mason, they've been mining a very productive vein. And you'll hear from them today about development and implementation of clinical proteomics for precision oncology applications and therapeutic drug development. Gentlemen, the floor is yours. Thank you.
Thank you. Thank you very much. It's a real honor to be able to present today. We're going to do a little bit of a tag team in our presentation, and we're really looking forward to telling you a bit about what we've been doing in our clinical proteomics laboratories, and hope that this will serve as a catalyst for more collaborations. And we'd love for everyone to think of us as, an external core, if you will, for some of the capabilities that we have. We have a lot of predicates for this implementation of some of the unique technologies and platforms that we have here. So again, we hope that this will serve as a catalytic event for us to be able to develop, individual scientifically driven collaborations like we've started with doctors Gannison and White and some of their own efforts. So thank you again, and we'll go ahead and we'll share our screen here, and let us know. Can you all see this?
We can, gentlemen. Thank you.
Thank you. So as you said,
you just need to go into presenter mode.
Yes, thank you. I don't know why it's not allowing me to go into presenter mode. Hold on for a second. Redo this. Great. Is that okay? Great. Thank you.
Are you seeing a change? Did you see the slide?
We saw the change. You're good to go.
You're good to go. Thank you. Thank you. So we'll begin by just talking about some of the fundamental reasons why we're excited about our some of the technologies in the field of proteomics in general. As they say, well, I guess DNA, the blueprints of the cell, it's really proteins that do most of the work. They're very often the drug target itself, especially for many of the targeted inhibitors that we use in the clinic. It's the proteins that are the drug targets. And of course, as we know, sometimes it's also protein biologics or therapeutics, antibodies, and even cytokines, chemokines, for example. So proteins are a pretty important aspect of biology, and they really tell us something about the functional activity of the cell. And as Dr. Labudis alluded to, even subproteomic components that are extremely important now, phosphoproteins, acetylated proteins, et cetera, are themselves becoming important biomarkers, predictive prognostic. So if we think about some of the roadblocks that we have to maybe implementing some of these proteomic technologies and really the dreams of molecular medicine and sources of innovation, of course, as we all know, individualization of therapy is now a massive undertaking, and the fundamental sea change in how we're treating patients is the individualization of therapy based on the molecular portrait of each patient's individual tumor. So we think about the overwhelming genetic diversity and cellular heterogeneity of cancer. A lot of times in the genomic side, it's what is the functional driver, what a causal driver of this patient's tumor. Proteomics offers direct functional access to the actual targets themselves, our proteins, and the actionability landscape can explode when you think about overlaying proteomic and phosphoproteomic information. And then strategies for discovering new therapies that block protein-protein interactions which are a hot area right now because of the potential for resistance development to be more difficult for PPI inhibition. And then really the functional proteome as reflected in the body fluid biomarker space is a hot area and has been a hot area for a while for uncovering new circulating biomarkers that can be used to detect disease like cancer as early as possible, where of course we can change the trajectory of the best. However, many of these biomarkers are extremely low abundance. And in fact, if you think about detecting diseases like cancer early stage, the actual stochastic input of the amount of biomarker should be very low, because you're hoping that you're discovering and uncovering cancer at its earliest stage where the tissue output is very low. And so we want to go, in fact, beyond that, even to cancer risk markers as well. And again, as I said, developing better ability to develop new drug targets and the drug targets of the future, especially in developing protein-protein interaction inhibitors. We know that many protein interaction sites can be flat, featureless, and even some of these have been classically considered undruggable. Things like RAS, for example. So we're now being able to take advantage of new technologies that we'll discuss and rapid mapping of the 3D amino acid sequence of these interaction sites using things like mass spectrometry to yielding new therapeutic leads. And Lance will talk a lot about this in the second half of the talk. But we want to talk and start with, in some ways, a philosophical underpinning of genomics-based precision medicine, which of course is the lead actor in our individualization of cancer therapies. The problem is, of course, is that many times these genomic alterations are low frequency. We in ourselves, and I'm sure many of us who are involved in molecular tumor boards or in programs like TAPR or NCI Match, many times we have patients' tumors that come through molecular profile and there's no match because that patient's tumor doesn't have an actionable driver uncovered by the targeted exome analysis, for example. And of course, the fundamental aspect is that because of the heterogeneity of disease and cancer, that in each cancer patient's tumor is different, that the frequencies by which any of these alterations, EGF, HER2, HER3, PIK3CA, as example alterations, ALK, ROS, these occur at relatively very low frequencies. So we sometimes miss on what actually is driving the tumor, and it's unknown which of the thousands of mutations are actually driving that patient's cancer. How many of us have looked at a report from a molecular profiling company and had all of these variants of unknown significance listed, and we're left scratching our heads on which of these could potentially be drivers. Now, we've had many investigators in the past try to expand our actionability space by looking at proteins and phosphoproteins especially, that because of their direct actionable functional performance in cancer biology really can increase the number of biomarkers that could be used to predict response to therapy. Active kinase signaling pathways mediate, we know, functional survival and growth, and we can start to think about these as patterns of phosphorylation. An EGF receptor, obviously one of the most classically described biomarkers for targeted therapy, the HER family especially, we know that it transduces its signal by a series of phosphorylation events in intracellular kinase domain, and that triggers cascades of signaling pathways driven by protein-protein interaction because of SH2, SH3 domains that are attracted by phosphorylation events that change the functional structure of those proteins and allow that to go on. Dan Hayes and others had showed actually for EGFR that in non-small cell lung cancer patients in wild type cancers that are not driven by EGFR mutation, for example, that measuring phosphorylation of EGFR in the pre-treatment biopsy samples and downstream AKT activation was highly correlative of response, even in EGFR wild type setting. Other investigators have shown the same thing with non-small cell lung cancer, that looking specifically at the pre-treatment biopsy sample, just the level of phosphorylation of EGFR was highly predictive of response. We don't give EGFR inhibitors routinely to cancers that are EGFR wild type because we focused mostly on genomic-based biomarkers of patient stratification. But in the background, we have an ocean of evidence that measuring phosphorylation can identify patients missed entirely by genomics-based approaches that respond just as well, if not better, than genomic-based drivers. So there's a lot of basis for measuring post-translational modifications in this instance with immunohistochemistry. And as Dr. Labudi said, that has its own issues, but with new technologies, we might be able to get around. And these are just some other background papers of looking at not just targeted therapies like EGFR inhibitors, measuring the direct readout of EGFR phosphorylation as a way to identify patients that will respond, but we can identify activation states in important pathways like mTOR and AKT that themselves may be not only able to predict response to AKT and mTOR inhibitors, but actually are fantastic and well-vetted biomarkers for predicting response to endocrine therapies, for example. And these papers by the Stockholm group on thousands of patients, over 1,000 patients, they looked by immunohistochemistry at activation of mTOR signaling. They found that in patients that have low AKT-mTOR pathway activation, that they're very responsive to tamoxifen. In fact, that tamoxifen benefit to these ER-positive patients in the adjuvant and neoadjuvant setting go out the door. And so those patients that have high levels of AKT-mTOR activation, this is not driven by PIK3CA or AKT1/2 or TORC1, TORC2 mutation. They're too rare to explain this. It's all at the level of activation. That level of tamoxifen benefit goes away completely. So there's lots of data that drive the potential for these types of biomarkers to increase our view of the targetable landscape. Now, Lance may want to get into a little bit of how the analysis of these at the cellular histological level can help as well.
Yes, I think that now that we see that phosphoproteins can provide important clues as to the pathogenesis of the tumor and its response to therapy, but in the past, this has been measured mainly by immunohistochemistry, which has low sensitivity for phosphorylated epitopes, and the tumor tissue is extremely heterogeneous. We would like to have quantitative measurements of how much phosphorylated protein in certain signaling pathways is present in individual cells within the tumor tissue microenvironment. And if you look at this example on the left, you can see that the PS6 and 4EBP1 phosphorylated versions, they're very heterogeneously distributed throughout the tumor tissue, and every patient's tumor is heterogeneously different with a different subpopulations of immune cells and host cells. How do you measure quantitatively the phosphorylation levels if you're faced with this dilemma? Next. And so we developed laser capture microdissection and reverse phase protein microarrays, which we combined together to allow us to microdissect directly under microscopic visualization. And now with the new digital versions of this, you just look on a screen and point out what you want. We microdissect only the cells that we're interested in measuring. And then those cells are lysed and printed on a slide, and Chip will tell you more about that in a minute. But the result is that we can actually map the activated state of signaling pathways based on using phosphospecific antibodies, microdissection, and the reverse phase protein microarrays. So we can say this pathway is activated by looking at the upstream and downstream measurements of that pathway, and we can do that in individual cell types ranging from the tumor cells, the stromal cells, or the immune cells?
And so our laboratory has spent the past decade or so not just inventing the underpinning technology, but of course, helping to get this technology out to the community, training in lots of different labs around the world. Trained the Mills lab down at MD Anderson. We trained several laboratories in Japan and at Baylor and around the country have come to us to learn the technique and implement it in their own laboratories. We've also spent a lot of time transitioning this from a research-based tool to a CLIA-based tool that can be used in real time for clinical decision-making or evaluating the performance of the markers in rigorous clinical trials. We've spent a lot of time in the past 1.5 to 5 years of really transitioning this technology to a CLIA lab. One of our special attributes of our proteomics laboratory is we're CLIA and CAP accredited. It's a fairly unique environment where we can transition research-based technologies or even biomarkers and validate them under CLIA and CAP and be able to use these under development in actual research clinical trials. One of the main attributes of the LC-MR PPA workflow is that it really is an unparalleled technology to study protein signaling in clinical and preclinical samples, where we can quantitatively measure hundreds and hundreds of phosphoproteins and proteins in microscopic amounts of tissue. In the laser capture dissected epithelial cells and the stromal cells next to it, we can do field effect work. We can look at infiltrating lymphocytic compartments. We can do immune guided LCM. And then this gives us the input to look at many different parts of different signaling cascades. Its quantitation, sensitivity, and multiplex capacity vastly exceeds multiplexed IHC or nanostring DSP or any type of multiplexed immunohistochemistry that provides spatial resolution. And again, we've been working on developing next generation platforms where we have developed this under CLIA. We have cut points, we have low and high controls like you would develop in an ELISA, say, where we can then interpolate or extrapolate a given patient's tumor sample value to a calibration curve, and this is what's taken forward in our clinical samples. We have lots of validation that go on all the time for the antibodies for the reverse phase array. We have over 500 currently validated, and the net result of this is that we can measure all of these types of proteins and phosphoproteins in a small part of a core needle biopsy from a clinical trial. We actually only get a part of a part because many of these, core needle biopsies are divided into compartments for NGS analysis and transcriptomic analysis, and we microdissect even small compartments of that. So I'm going to tell you a few vignettes of how we're using this in clinical trials today to give some examples of the utility. So one of the clinical trials we're very much involved with, our laboratory is a protein biomarker engine for the I-SPY2 neoadjuvant trial. It's a very well-known neoadjuvant trial by Laura Esserman's group at UCSF and many participating sites. This is one of the first adaptive design trials that Don Berry developed and deployed, where depending on the patient response by MRI, you actually mathematically bring in patients in a weighted way to get the different drugs depending on response, and drugs that work well graduate and those they don't are kicked out. We've deployed our technology and workflow microdissection reverse phase array in a number of different settings for I-SPY. And in one of the very first settings, we looked at the concordance between HER2 levels that we obtained from these breast cancer cases with central FISH and IHC to be able to at least credential whether or not the values we get, how well they match an orthogonal FDA-approved approach. In this instance, we have central FISH data, which is shown on the x-axis of ratio of HER2 to CEP17. Of course, anything above 2 is considered amplified. And then our quantitative HER2 value is the y-axis. And in general, patients that are HER2 unamplified in red, HER2 amplified in green, that are separated by FISH, you can see you have a nice concordance between patients who have FISH amplified or higher HER2 by RPPA, and the patients that are HER2 negative by FISH are low by RPPA. However, when we looked at these patients, and this is for total HER2, when we looked by phospho-HER2, we found a fascinating cohort that emerged. So these are the same patient cohort. And now, even though we had very good concordance between central IHC, FISH, and RPPA, we looked by phosphorylation. We found a group of patients that was FISH negative, but had phosphorylation of HER2 at levels that were equal to those seen in the FISH positive population. So these patients in red in the circle are all FISH negative, but they're phospho-HER2 positive, if you will. To put this into a functional context, none of these patients would be getting HER2-directed therapy, even though they actually have high levels of that drug target activated. These patients are not HER2 amplified by genomics. They're not HER2 high expressors by transcriptomics. They're not HER2 mutated. This is an entirely different cohort of patients that's highly activated and yet genomically negative, if you will. And we published this finding in Clinical Cancer Research and validated it in a large study set with collaborators in Germany, where we found, again, this subset of patients that are FISH IHC negative but phospho-HER2 positive. They also have dimerization, with EGFR and downstream SHIC, FAC, and STAT activation. So we saw basically evidence that the pathway was activated. Now, this was an observational study, and again, not explained by any underpinning genomic mutations because the prevalence is so low. And while this was observational, we had an opportunity in I-SPY2 to test the functional significance of this HER2 phospho high group that was HER2 negative by genomic alteration means of measurement. And that was in the drug neratinib that was actually given to HER2 to triple negative breast cancer patients because of the potential for EGFR kinase activity with neratinib as a pan-HER inhibitor. So we knew from our previous work that we had this cohort of patients that was phospho-HER2 positive, and we speculated and actually made a qualifying biomarker prediction in I-SPY2 that said that patients that were phospho-HER2 positive in the triple negative group would respond to neratinib. And we made that as a qualifying biomarker prediction. And as you know, again, neratinib targets the intracellular domain of it's a true TKI that targets the kinase activity of EGFR, HER2, and HER4. And in a paper that we published in JCO, Precision Oncology, we basically found that we were right. And those patients that had high levels of HER2 activation and EGFR activation, this is a two-way scatter plot of HER2 activation on the y-axis and EGFR activation on the x-axis. The responders in yellow basically all clustered in this top quartile quadrant here of high levels of EGFR and HER2. This is not seen in the control arm. And the prevalence of this HER2 positive, EGFR positive population was about 43% of triple negatives that have this phenotype. And these patients had a high PCR rate of nearly 80% and a probability of success in the phase 3 setting for the selected cohort of 95%, which is what Don Barry uses to select drugs going forward in phase 3. So this is a cohort of patients that we're now trying to prospectively validate. We're working with some drug companies that are very interested in identification of this new triple negative cohort, again, missed entirely by genomic-based approaches that have phosphorylation of the drug target itself activated, and we now know are highly correlative with response. We found that this EGFR HER2 activation phenotype was not just important in the triple negative group, but also important in the HER2 positive group. We were able to test this in I-SPY2 with TDM1 pertuzumab and pertuzumab trastuzumab in ostensibly HER2 positive patients. So these patients were already supposedly HER2 positive by FISH and IHC and were brought in to receive TDM1 pertuzumab and pertuzumab trastuzumab therapy. And what we found again is that, and it's the same type of scatter plot of activation of HER2 and EGFR. You see a nice diagonal. So there's coincidence of activation. You can see patients that have higher levels of EGFR activation have the highest HER2 activation. And this continues to go down the line. The patients with the lowest levels of EGFR activation had the lowest levels of HER2 activation. What was nice, though, was that when you overlaid this on pathologic response, you got the same type of phenotype, even though these are already HER2 positive by IHC and FISH. Those patients that are HER2 positive are destined for non-response, are localized down in this bottom quadrant, and all the patients that have high levels of HER2 and EGFR are all destined for response. We get a nice separation, if you will, of this of dichotomization by a cut point of being able to pre-select patients who would be potentially could be deaccelerated for therapy, or patients who are destined for non-response could be accelerated for therapy. And this included people who were 1+ and 0 by IHC and 1+, 2+ by IHC. So the hope is that we would be able to use this type of finding to further validate in new cohorts and potentially use this to accelerate or deaccelerate therapy by risk stratification. And this is something that we are proud that we have been able to receive funding from the DoD to collaborate with Dr. Sridhar Ganesan there to take advantage of the Rutgers cohort. We'll be working with Sridhar to validate our cut point in retrospective sets and take this forward in prospective studies with him as well. And we hope that our HER2 pathway activation is a new biomarker stratification, I think, ecosystem, where by looking at the functional activation state, we can gain enough sensitivity and specificity to be able to identify patients who are destined for non-response and get them accelerated therapies and new combinations, and patients who are destined for response, potentially we could deaccelerate, reduce unnecessary toxicity from chemotherapies that could be taken away, but we still get good response. So again, we're really happy to be able to work with Sridhar for this important prospective work. And then lastly, I want to tell you about a trial where we are not just taking this for retrospective biomarker discovery and correlative science analysis, but we're actually using this data for use for treatment decision use in molecular tumor boards. And this is sponsored by the SideOut Foundation for metastatic breast cancer patients who have progressed off of standard of care therapies that are undergoing multi-omic profiling by multiplexed IHC, genomics by Caris and Foundation Medicine, and then our laboratory doing the reverse phase array analysis on a prescribed drug targets that are being done in real time, that information goes to molecular tumor board. So again, since we've developed this under CLIA, we have calibrated the assay for many drug targets where we have predicate population data. We have low and high controls that are printed on every array. What that allows us to do is identify a referent sample set of patients, tumors that are mimic the intended use of our population. We can take that patient referent range and compare that to an incoming patient sample, where we can then determine the levels of activation and then generate a report for the levels of activation for many different drug targets and downstream signaling. This report is generated within 5 to 7 days from beginning to end, from laser dissection all the way through reverse phase array analysis. And this is done in a very MTB-friendly way. What we were able to do is compare the progression-free survival interval for these heavily pretreated metastatic patients that were selected for molecular targeted inhibitors based upon our data compared to their previous PFS. And this is work that Dan Van Hoff had kind of pioneered for, which basically a score where we are looking for a 30% increase of PFS based upon our molecular profiled informed therapy compared to the prior therapy. And we found that in our our trial, which actually met its primary clinical endpoint. We had over 50% of the patients. Again, these are heavily pretreated patients with an average of 5 therapies that have failed the patient beforehand. That the PFS on the molecular informed therapy from our assay was at least 30% or greater. In red is the preceding PFS, and blue is the PFS interval on our molecularly informed therapy. We've been able to look at the specific drug targets, look at the specific concordance with other genomic drivers, and we're taking this into independent validation with a larger set with CytOut based upon these findings. So one of the hopes that we have is a reclassification of cancer by functional protein pathway activation, instead of thinking about classification by location or type, thinking about reclassification on, alcomas or sarcomas, but not sarcoma, but sarcomas, raphomas, mtoromas. And what we're thinking about is designing types of umbrella and basket trials that can test that theory out. In the future, we hope that a pathologist will be able to get a individualized protein pathway activation report that would accompany every patient's pathology report that tells about what circuits are activated functionally in that patient's tumor, that can help the medical oncologist and the pathologist help to construct patient-tailored therapies of the future. So with that, I'll end that part of the talk, and we'll be taking questions at the end.
Now we're going to talk about biomarker discovery using proteomics and special new technology, followed by protein-protein interactions as new sources of drugs. So we all want to have a blood biomarker that we can measure, and it will tell that a patient has a pre-metastatic early stage cancer. Unfortunately, that has not happened in the past. And even though it's a dream of biomarker scientists, it's been extremely difficult to accomplish because blood-borne biomarkers exist in very, very low concentration. Imagine just a tiny tumor, a few millimeters in size, how few biomarkers are emanating from those cells, and they're then immediately diluted in the entire blood volume. And once they get in the blood, they're obscured by abundant proteins like albumin and immunoglobulins, which they stick to. And then they're rapidly degraded by exogenous enzymes in or outside the blood when they're collected. And we have a very hard time, even if we discover some new candidate markers, we have a very difficult time getting specific ways of detecting those markers. And so we set out under NIH funding to develop completely new technologies to solve this problem, how to increase the specificity and, most importantly, the sensitivity of biomarker discovery so that we could see biomarkers down to 0.1 to 10 picograms per milliliter. And we accomplished this, and it's been used for infectious diseases and cancers and head trauma, and even honeybee, looking at proteins involved in colony collapse of honeybees. Use it for all these different general methods because this method can allow us to harvest and capture very, very low biomarkers that cannot be measured by any other method. And what the technology consists of are smart nanoporous nanoparticles, which have 4 functions at which they accomplish within minutes in one step. They sequester and molecular size sieve and affinity capture the analytes of interest in the body fluid after we just dump in these nanoparticles. EM pictures of them are showing up in the upper left. They concentrate all of the solution phase target molecules into a tiny volume, and then they completely protect the harvested proteins from enzymatic degradation after they capture them, and they can be made to exclude albumin and high abundance proteins that get in the way of the analysis. And the way these work is that they have immobilized special chemical dyes that we've discovered that bind proteins at many parts of the surface of the protein with extremely high affinity. As you can see in this specific example, small molecule organic dye, that we've looked at hundreds of different candidates and synthesized some new candidates ourselves. The binding of a protein to this dye is incredibly fast, and the on rate is incredibly fast, and the off rate is very, very slow. And so when we dump the nanoparticles into a body fluid, they sequester what we're interested in having them capture. Then we can spin it down into a very small volume that then concentrates the what we've captured for then going into mass spectrometry or any other immunoassay method or any type of analysis. And so here's an example of application of this to discover of low abundance biomarkers that are specific for early stage breast cancer and differentiate breast cancer from other types of cancer. So based on a US-Italy collaboration, we obtained a very large number of blinded samples from breast cancer patients, but also from colon, prostate, and ovarian, and melanoma and lung cancer patients. We looked for specific biomarkers using the nanoparticle harvesting, followed by mass spectrometry, and we ended up identifying a constellation of biomarkers that were in the cancers, but not in the controls, including disease controls, but more importantly, that were only found in breast cancer, but not found in other cancer types. So we came up with this is the number of validated in the first phase of the study biomarkers for other cancer types compared to breast cancer, and we found a set of specific biomarkers for breast cancer, which we then developed an MRM method, which is a quantitative mass spec method for looking at those specific markers. And what we found is in an independent, separate, blinded study that we could detect 1A early stage small breast cancers by a panel of markers, and the sensitivity and specificity analysis and the ROC curves came out very good with in some cases 100% sensitivity, in some cases 92% specificity. But when we combine those markers together in a panel, then we had the highest level sensitivity and specificity, and very good ROC curves. And the discovery that we made that we were completely unexpected is the markers did not seem to be coming, at least these high predictive markers didn't seem to be coming from the breast cancer cells themselves. They came from stroma-related proteins. And these proteins, we know that many of these proteins, stroma proteins, are found in the stroma of breast cancer. And so what we might be picking up here is the completely different stroma that any pathologist notices when you look at breast cancer. The normal stroma around normal breast ducts is very loose and has very low amounts of some of these proteins that we find instead in the high density stroma, reactive stroma around invading breast cancer cells shown here. So we think that we're detecting early stage breast cancer here because we're looking at the change in the surrounding stroma that any pathologist sees at the area of even microinvasion. We've also used this same high sensitivity method to look at lung cancer field cancerization risk markers. So the concept here is that as the field cancerization takes place in the bronchial tree preceding lung cancer, that we might be able to see changes systemically from that bronchial epithelial evolution towards carcinogenesis, towards the tumor. And so we obtained studies from the large physicians' health trial, where we were able to collect samples at many different times before the lung cancer developed, And we were able to then discover in smokers and never smokers and former smokers even, a series of fascinating proteins. Here are the categories that were involved in airway modification, airway remodeling, lung morphogenesis, pulmonary inflammation, chaperone-mediated autophagy, and different categories of biomarkers in a blinded study where we confirmed these findings were found between the never smokers and the smokers, and the smokers compared to the former smokers had similar markers. Again, I want to emphasize these are risk markers that are predicting future lung cancer 2 to 5 years from the time that we're obtaining the sample. And then we found a very high percentage of these markers that we found in the blood of the patient were also found, 82% of them, in the actual bronchial tissue that we obtained at surgery near to the tumor. And that was different than what we found far away from the tumor. So that we believe that our nanoparticle technology for discovering biomarkers is another really good type of collaboration that we can offer to colleagues at Rutgers. And I'm going to end with our another technology we developed to study protein-protein interactions. And when I talk about protein-protein interactions, I'm not talking about just who is stuck to whom. I'm talking about where they're interacting. What's the amino acid sequence of their binding domains together? Very hard to find that out by crystallography, because it's very hard to get a crystal in the first place, even harder to get a crystal structure for interacting proteins. And the method that we develop allows us to use a combination of special dye chemistries and mass spectrometry to immediately get a readout of the amino acid sequence of only the place where the 2 proteins or 3 proteins or unlimited number of proteins are interacting or touching each other. And why is this important? Because all types of signaling in the cell, from transcription factors to membrane-associated interactions to complex movement of proteins within the cell, traversal of the cell membrane, it's all based on protein-protein interactions. Gobs and gobs of targets for therapeutic development. But it's been thought in the past it's really hard to find those protein interactions, particularly if they're not just a simple drug, a simple enzyme substrate pocket. When it's a flat surface, it's very difficult. So our technology called protein painting, which is very simple to do, you take the native proteins when they're bound together, and this can be not just purified proteins, but mixtures of proteins. You mix them with special new dye chemistries that we find that coat the surface of the proteins, but do not alter the 3-dimensional shape of the proteins. And then we dissociate the proteins and subject them to typical mass spectrometry. The dyes that I mentioned, the special chemical, small organic chemical dyes, they do not come off. And now we have the denatured proteins after they're dissociated, and in the regions of this complex 3-dimensional protein where they were previously touching each other, there's no dye molecules. And now if we use trypsin to cleave this out, we can find that after we trypsinize it, only the regions where the dye is not covering up the protein is assayed by the mass spectrometry. So direct output of the mass spec is only the binding sites between those 2 proteins. So now I'm just going to go through quickly some examples of how we've applied this to discover new hotspots of interaction between different cancer-relevant biomarkers. For inflammatory cytokines, we took on the challenge of discovering how the 3-way proteins between the IL-1, its receptor, and its accessory protein came together, and we used protein painting and found a specific biomarkers, specific hotspots on the receptor, the accessory protein, and these are all marked in color here. So now we had ways to potentially develop a drug against that. So we developed a drug against one of the most high energy hotspots that we found. And it turned out to be a beta loop type drug that was multivalent, hitting the complex at 2 points, and it was quite good of an inhibitor. And we tested its ability to block the protein-protein interactions in vitro. And then we tested it, and it was very effective in inhibiting interleukin-1 signaling in a mouse model in vivo. We also have used this to look for the first time ever at the phosphopeptide binding domains in BRCA1 protein, because that has a big impact on the function of the BRCA1 molecule. And there might be a way to use these types of phosphopeptide inhibitors as a means to sensitize the patients to PARP inhibitor. And we verified this with a collaborator. In fact, we found binding sites that were completely unexpected, and we verified them by putting extra tags onto the phosphopeptide. We also use the protein painting method to find new interaction domains in the hippo signaling pathway. As you know, the hippo signaling pathway is a very important type of oncogene or oncoprotein, and it's involved in cell size, contact inhibition, cell migration, angiogenesis, and a host of other factors. And it's very important in regulating drug resistance. So we used our method to map specific binding sites, specific hotspots on all the major interaction components of the hippo pathway as shown here. And then we found some really interesting new hotspots discovered that were not known before, because those parts of the protein were never crystallized. And we also, some parts of the protein we don't know what their final structure is, but we're still able to find that. So this gives us all sorts of new directions for inhibitors for the hippo pathway. Very exciting topic. We can use our method to look at gigantic transcription factor complexes, and we've done that very successfully with the BAF nucleosome remodeling complex, and we can find all the proteins in the complex that are on the outside of the gigantic complex, and those are the ones that can be mutated, and they're involved in cancer, as you know. So, and our results confirm previous data and will be used to generate better models of the complex for drug development efforts. Lastly, we've developed a new inhibitor for PD-1, PD-L1, well known to all of you as a very important means for the tumor cell to resist recognition by the immune system. And so we found a hot spot that was never really identified specifically before between PD-1 and PD-L1. We made peptides, mimics of it, that showed very good dose response curves. And now we have a new peptide potential peptide small molecule inhibitor of the PD-L1, PD-1. And this is compared to the antibody, Keytruda, this is a very tiny molecule. So we think that'll be much better for immunotherapy of cancers. So in conclusion, the protein painting is a means to non-covalently coat proteins in native conditions. In a few minutes, you can, with a resolution of 3 amino acids, you can find only the binding sites between any number of proteins, or you can study protein folding this way. And it uses standard mass spectrometry instruments and analysis software. So we think this is another example of an exciting potential source of collaboration with our Rutgers colleagues. So in conclusion, we're very thankful to have this opportunity. This is our team at George Mason University, and just some examples of some of the grants and the agencies that have supported us. Thank you very much.
Thank you, Chip. Thank you, Lance. it was a tour de force, very, very impressive. So we have time until 2:15 for questions, and you may either put your question in the chat or put your hand up, however you would like to do it. And I'm looking at the chat right now, and I'm ready to recognize anybody who wants to ask.
Looks like
questions.
Stephen Zhang has his hand up.
Okay, Stephen, please go
your
ahead.
partner in crime.
Yeah,
Hi,
my
this is Stephen
better half.
Zhang, from Rutgers Cancer Institute. Great talk, exciting work. So I have a question about protein painting. So that's a very exciting technology. So I was wondering, the labeling is more of on the periphery of the interface, or if the small molecule can penetrate into the heart of the interaction?
No, let me see here. So the dyes that we have discovered, and this was work of our chemist, Amanda Heyman and Dr. Lucchini in our group, the dyes that we find, they bind to the outside surface of proteins at different points, and they seem to like the regions where trypsin also recognizes a cleavage site. But they're looking only at the solvent accessible regions of the complex. They're excluded from the solvent inaccessible regions, which is where the hotspots are. And in fact, this allows us to paint or coat all the trypsin cleavage sites on the outer, on the proteins, all around all sides of the proteins, but not where they're touching each other. So now when we dissociate the proteins, they're coated with these dyes. We can linearize them. When we chop them up with trypsin, the only available sites for the trypsin to hit are the inner foldings of the protein, which weren't accessible anyway, which we can control for those by looking at before and after painting those proteins themselves. But more importantly, we see that with before and after we paint, we see a pop-up of the amino acid sequence only of where the proteins were touching each other. And that's how we discovered all these markers, and then we're able to validate them. So for example, when we're doing it for sickle cell, we're looking at how the hemoglobin molecules bind together. And we can even follow that over time and see, like a zipper, where the different amino acids are talking to each other and touching each other as the hemoglobin molecules form that polymer. We know that the dyes as shown here do not change the conformation of the protein. This is a Heyman et al. JBC paper. In fact, they even protect the protein a little bit from denaturization. But if we painted those proteins during different stages of the folding of the protein, it would block the folding just as it blocks the interactions, because it sits on the surface in the solvent accessible phase. So does that help you?
Yeah, that's a great.
So we know why. We know how these dyes bind. They have two like hydrophobic anchors and a central region that, it's a combination of hydrophobic pi-pi interactions, anionic, cationic interactions, and then the number of binding sites on any protein is linearly related to the size of the protein, because it binds all over the protein.
So if we would like to collaborate with you, so what would be the mechanism? Should we contact you directly, or?
Sure, just contact me or Dr. Petricoin. We'll set you up with the collaboration. The way it usually works is you don't have to. Ideally, you'd want to know what the amino acid sequence of your proteins are that you're interacting, and then we would see. We could, because we get both sides of the interface. We can match it up and make a model of how we think they're interacting, even if you don't have a crystallography three-dimensional. We're really good at looking at antibody binding domains. Where does an antibody bind by this method? So you would just we could teach eventually want to teach them how to do this.
Yeah.
Your mass spectrometry group, how to do this. It's very simple. You're just going to mix the dyes with the protein complex that you have very rapidly. Then it just takes a few minutes. You put it through a spin column so that your bound proteins that are coated with dyes come out the bottom, and the free dyes are at the top. You just take that and you process that by standard mass spectrometry and what comes out of the mass spec is only the binding domain amino acid sequence.
But definitely, Ed and Steve, they have our emails. We're happy to provide them to the group if we're given a group participant list, and away we go.
Yeah, you just send us your proteins, or we'll teach you how to do it.
Thank you very much, gentlemen.
Data map, we prefer that to do that map that I showed you. We have where we say where on the amino acid sequence of your protein where the binding sites are. Now remember, this is not like even though there's a complex three-dimensional shape of the interacting proteins, we're not looking at the linear interaction. We're looking at the complex three-dimensional, who's touching whom. That's what we get out of this.
So, gentlemen, we have a follow-up regarding the painting, and Harmeet Kaur from the Protein Data Bank asks how the trypsin cleavage sites are specifically targeted. Is there a covalent adduct formation with lysines and arginines?
It's not covalent. No, it's non-covalent. But the dyes that we selected after screening hundreds and hundreds of dyes prefer probably from a more hydrophilic interaction, the regions near the trypsin cleavage sites. We also have other dyes that we're using with other enzymes to chop up the proteins, because even if you have a big trypsin peptide that's produced, you might miss the areas in between.
Indeed. So other proteases.
Right, so we have other proteases, but they're not as specific. Trypsin is still the best specificity for accuracy for predicting the outcome.
Thank you. And I'd like to recognize Sridhar Ganesan, one of our speakers this afternoon, who wanted to ask a question.
Yeah. Hey, great talk. Just a quick question for people in general. So in terms of how much material do you need for the phosphoprotein analysis in clinical specimens? And in terms of separating signaling in stroma versus the tumor cell, have you guys looked for downstream activation, for example, downstream of PD-1, to see if PD-1 expression is also associated with evidence of downstream activation of signaling pathways? And that might be a better marker of for immunotherapy as well.
Yes, so from a operational technical standpoint, obviously one of the attributes of the RPPA is its sensitivity, and so great for clinical specimen analysis. We routinely microdissect just about 10,000 cells. Generates enough of a lysate to look at 150, 200 proteins, phosphoproteins. To put that into kind of a context, and we can do this from FFPE as well as from fresh frozen. So we do not require fresh frozen. We just require freshly cut FFPE sections. You don't see the same degradation in phosphoproteins that you may see ostensibly by IHC, but we think a lot of that is antigen retrieval issues, not degradation. But notwithstanding that, we routinely get 5 to 7, 8 micron sections of even like an 18 gauge core needle. And that's enough to, depending on the tumor cellularity, in general, 20–25% tumor, that amount we can look at hundreds and hundreds of signaling proteins. We get much less, and we can still look at 30, 50 phosphoproteins and proteins in our targeted kind of CLIA analysis. We may only get a few sections of an 18 gauge core needle. We also working with cytospins. So we can we have a collaboration with Vicky Seawald's group at City of Hope, where they're looking at retroperi-alveolar fine needle aspirates, and we're doing cytospins of RPFNA and microdissecting the epithelium out of that for the input. So very microscopic quantities. And then yes, we actually are looking at an I-SPY with pembrolizumab is being given. We're looking at stromal immune checkpoint and stromal signaling, phosphorylation of ZAP70 and other LCK and other downstream cytokines, chemokine signaling, and correlating that with PD-L1. We have some very intriguing findings we are hoping to verify in the Pembro ×8 arm that we would be hopefully submitting to San Antonio this year.
Thank you, Chip. We have a question from Greg Critchlow from the Protein Data Bank.
Great.
Greg, would you like to unmute?
Thank you. I was wondering, you mentioned that you had you were able to look for biomarkers for breast cancer specifically, and it turned up there were mainly stromal markers. I was wondering, one, do you think that's mainly because you have, generally speaking, some of the same molecular drivers within the tumor, as was mentioned in the first part of the talk, instead of classifying as breast or prostate, et cetera? You have the same drivers that may be in different tumor populations from different organs.
Yep.
And then I was also wondering, being that you found stromal markers specific for breast cancer, does that same specificity hold up when you look at metastatic sites? And do metastatic sites, do you see the same stromal markers that came from the original breast stromal site?
Great question.
Yeah, so with regard to the tumor cell derived markers, yes, because we're looking, that's a very good point, because carcinoma cells, these were other types of carcinoma cells, likely they have similar kinds of changes that are carcinogenic. And so they're going to not filter out as being unique for breast as much. We expected to find some breast specific markers. And in a new study, we're looking for markers that are associated more with coming directly from the breast epithelium in a suspicious mammogram study. But we found that, but in this study, I think we're finding the stromal markers are popping out because that differentiates the type of markers that we're finding for all carcinomas or other types of carcinomas and breast cancer. And you know, as you look at the stroma of breast tissue, it is very, very different than the normal breast epithelial stroma surrounding normal breast epithelium. So that could be the reason, exactly as you state. With regard to the metastatic tumors, we don't have as much data on metastatic tumors, although we've done some studies in the past with metastatic cancer. We're trying, and the reason is that we're focusing mainly on the very early stage breast cancer, or even the pre-malignant lesions, such as in the lung cancer, bronchial epithelial field effect, or even carcinoma in situ of the breast. Because we have such high sensitivity, we're trying to take it where it's really needed. And we're also looking at the microbiome, which is fascinating in what we're finding, because our method allows us to be so sensitive that we can sequence thousands of the proteins coming from the microbiome.
We have seen, interestingly enough, on the cell signaling side, not necessarily on the circulating kind of biomarker side, that underpin that part of the talk with the stage 1A breast cancer markers that were stromal derived. We actually do see some evidence of both seed soil influences on the signaling architecture within the metastatic lesion. We have some papers we'd be happy to send you where we looked at, breast cancer and its signaling architecture, both in the tumor and also in the metastatic microenvironment for, say, liver metastases versus brain metastases versus lung metastases. And of course, we're really interested in collaborating where we have these longitudinal phylogenetic patient-matched primary metastases study sets that can become very interesting to study from a clonality standpoint. But we're finding evidence that certain signaling architecture is somewhat starts to coalesce depending on the site of metastases. For example, we published a paper that in the hepatic microenvironment for liver metastases from colon, breast, other solid tumors, they actually started to look more alike in the hepatic microenvironment than they actually did in the primary setting. So it may be that the soil starts to select out certain signaling pathways that are needed to survive in, say, a foreign soil. So it's a fascinating, kind of question, and we're very, very interested in collaborating with folks who are
want
interested
to
as well.
emphasize in the side out trial that we mentioned, those are biopsies of the metastases itself. That's not
Exactly, Lance. So
primary,
may
primary
I
tumor.
interrupt, please,
Yep.
and call on David Axelrod from the Department of Genetics to ask the last question.
Thank you.
David, are you still there?
Yes, I just put it in chat. So the question
go ahead.
is on the predictive biomarkers for lung cancer, I believe it was, from plasma. So you identified something like 5 biomarkers. How did you identify those, and how did you combine information from the 5 different biomarkers?
So those are from work.
And
The
if you could cite a reference, I'd appreciate it.
Yes,
Yes, send it to you. Yes.
we'll send it to you. So we use the nanoparticles to harvest the plasma of patients in the physician's health study, where we had samples given that were collected at different times before the patient developed lung cancer. Then the lung cancer was, definitively identified by pathology. And these patients were known to be former smokers, meaning they stopped smoking, current smokers, or never smokers. And then we looked to see if we could, in blinded fashion, whether we could find markers that correlated with whether that patient was going to get lung cancer in the future several years later. We also did a confirmatory separate study, not of those same patients, but different patients, where the patient was undergoing a lung cancer lobectomy, and we microdissected different places of the bronchial epithelium close to the tumor and far away from the tumor. We also used the nanoparticles to harvest the markers coming from, this case, the tissue, not the blood, to see if we could show that anything that we found in the blood from the first study actually came from the bronchial tree, because we think that the cancerization, field cancerization, early stage effect for lung cancer might have a much bigger surface area to generate markers for prevention or diagnosis or risk. And so we found a lot of the same markers that we found in the blood were also in the bronchial mucosa. And so that's giving us indication there might be some functional role for them. So that's the explain a little bit more to help you.
Thank you, Lance. So let's
Happy to
thank.
send you, David, the paper.
Yeah, thank you. Let's thank our speakers once again for a great talk.